Okay, let's get going. So this is lecture 3.4, the fourth lecture of Unit 3, and we're delving into MHC. So I've been hinting at this, dropping little notes of MHC basically since the week 1 of the course. So I'm sure you guys have been on the edge of your seat waiting to figure out what's going on with that. So this is the major histocompatibility complex, that self-marker that I've been referring to all of this time. So this is a genetic region encoding for membrane-bound proteins that present antigens to T cells. Also establishing self versus non-self. If I don't have MHC proteins off of my cellular membrane, then that is a big red flag. If that protein is messed up or not present, then that's a solid hint that that cell is either corrupted or not of my own. In humans, we call these human leukocyte antigens, HLA. So that's what the MHC code's for. It is found on one chromosome's short arm, chromosome 6. You don't need to know the P21 nonsense, but chromosome 6 is just on one chromosome. I find that kind of cool. It is highly polymorphic, tremendous variability in population. So if we harken back to the leukemia discussion and the bone marrow transplant discussion, ideally we have a HLA, an HLA match. That way, whenever we donate all of that stem cell, that blood cell population, we don't have an immune response. But it is very, very variable. Very variable. So that's why I said like maybe a sibling would work better, because you have a 25% chance that you match with your sibling. Okay. Let's press on here. What are the functions? So these MHC compounds that are hanging off of your cell membranes present peptide antigens to T cells. So they're basically like a silver platter that you might pluck some hors d'oeuvres off of. They're saying, hey, is this your peptide antigen? And remember, it's 1-to-1. If that T cell is born and bred to fight that antigen, it'll take that hors d'oeuvre right off the platter and start an immune response. Again, self-recognition. These immune cells learn which MHC is the self, which is ours during development. So that is that whole idea of preventing autoreactivity, that negative selection checkpoint, the second checkpoint that we talked about in both T cells and B cells. During that time, they're saying, oh, that's on all of this guy's cells. Why would I react to that? All right. If they do, apoptosis. So that is the big deal here. We recognize ours is ours during development. Immune coordination is a big thing too. So this determines which arm of the adaptive immunity is activated. So if it's CD4, that's going to bind MHC2, and CD8, MHC1. So remember, this is, how should I say, a surface level explanation. It is a little more complex than this, but you could think of this as, extracellular intracellular there is overlap technically, so that's why I said, proceed with caution, but that is a fine way to start thinking of it, because the CD8 response is more so dealing with viral infections, dealing with tumorogenic cells, while the CD4 response is more so for things outside of the cell, like bacteria and whatnot. All right, let's break it down. What do I mean by MHC1 and MHC2? Let's start with MHC class 1. So this is the look that we're going for. 3 alpha chains supported by one big old beta chain. In that alpha chain right up here, we have a peptide binding groove. Right, kind of crazy. It's only really 8 to 10 amino acids long. And that confers enough specificity. That can be an antigenic peptide that our body can recognize, which is kind of crazy to me. Not a lot of peptides. Or, I'm sorry, not a lot of amino acids long. Then we're anchored down by this transmembrane tail. We have this structure here. It is expressed on all nucleated cells. MHC1 is everywhere. Okay? That allows for immune surveillance of virtually all tissues. Think about how advantageous that is. Since I'm expressing it on all nucleated cells, I'm expressing it on all cells capable of getting hacked by viral machinery, right? Viruses can't reproduce on their own. That's why they are not technically alive. They have to use a host. They use a host's DNA replication machinery. We find that in cell nuclei. So excellent that I have that defense in place. Okay? What is the antigen source typically for MHC1? So this goes back to what I was talking about over here. Typically, endogenous antigens. So endo means within the inner layer. Viral proteins inside infected cells, mutated or misfolded proteins. The 3D modeling of a protein confers its function. Right? None of them are cubes. I can't draw an actual 3D protein. But one 3D conformation might be fine, and one might not be. So that misfolded protein might just be problematic in and of itself, and that can cause a response. And peptides produce from a proteasome degradation. So that's basically just saying all of the stuff that's getting broken down and the trash, the stuff deemed not of my origin or not helping the inside cellular machinery, that has the likelihood of getting presented. So we put that piece right here, that little nougat, and then we say, hey, is this anybody's? Does anybody want to react to this? Is this thing a problem? So that's how that works. We maybe take a piece of viral DNA and just present it on the membrane and say, hey, does anybody know how to deal with this? Because this was in my cell, and I'm not sure if this is okay or not. And then hopefully, a little CD8 T cell comes along, and we get a good binding there. That's the idea. All right. Moving on. So let's talk about how we put that little virus nougat, how we put that on the MHC. So all this stuff is the same. But we're talking about the processing pathway now. So we get protein degradation by the proteasome. So that's just one of your organelles that breaks down proteins. That peptide is then transported into the ER by something called TAP. MHC1 is built in your endoplasmic reticulum, the ER, the protein fashioning center. And then that whole complex travels to the cell surface in hopes that it gets recognized by a CD8 T cell. Ooh, we forgot something here. There we go. All right, so let's walk through that again. Step 1, protein degradation. So here I just have a random protein floating about in the cell. Then I have a proteasome down here. And it breaks down that protein into its constituent little amino acid chunks, little antigen chunks. Then we're transported in to the ER by tap. So down here is the ER. And I load this antigen chunk onto MHC1. So I built this complex in my endoplasmic reticulum, because after all, it's a protein, and endoplasmic reticulums make my proteins. I take that antigen complex, that little nasty thing that I broke down here, and I load it onto MHC1. Then we just pop it off into a little vesicle of its own, and that traffics to the surface of the antigen-presenting cell, or any nucleated cell. We are MHC1 after all. And now it's posted up on the membrane. What I have next to it here, right, that's my T cell receptor with my alpha and beta chain. And then since I'm a CD8 T cell, we also have the secondary receptor hanging out, making sure that activation goes well. All right. And then technically you have your CD28 and B7, but don't worry about that for here. Yes, it's still going on, but I just want you to get this pathway down first. Protein gets broken down into antigen-sized chunks. TAP brings me into the ER, the endoplasmic reticulum, loads that antigen onto MHC1. MHC1 pinches off and assimilates with your with your membrane, with your cellular membrane. So now it's pointed out into the world, into the extracellular space, hanging off the membrane, hoping that a surveying immune cell will deem it as problematic. So that's that CD8 MHC1 kind of process there. Press it on. MHC2, a little more boring. We just have these pillars. Kind of looks like the T cell receptor, honestly. Just these two shoots with an alpha chain and a beta chain. The top of the alpha and beta chain are going to form that peptide binding groove, just like the peptide binding groove we had in MHC1. Who expresses these? These are found in professional antigen-presenting cells. So are APCs. I should have had this covered up so you'd have to remember. But these have come up in each unit now. So, yes, important for test stuff, but if they're coming up in 3 units, then that just means important in general. So a good concept for you to take away. These 3 cells are my antigen-presenting cells. My dendritic cells, my macrophages, and my B cells are going to express MHC2. It can be induced in other tissues during inflammation. So, remember, it's on chromosome 6, MHC1 and MHC2. All of that stuff you need to make those compounds is right there. So, in times of stress, maybe a transcription factor makes its way into the nucleus and says, hey, we need more soldiers on deck, more antigen presentation on deck. We can upregulate MHC2. But that's a little in the weeds. This you should absolutely know, though. MHC2 is on my antigen-presenting cells proper. The source is typically exogenous, so extracellular bacteria and soluble extracellular proteins, so things on the outside of the cell. What's the processing pathway like? Ooh, I missed my A here. That's kind of funny. Let's make that red. There we go. Antigen is eaten up by phagocytosis. So, phagocytosis. It brings in that content into the cell from the outside, from that exogenous world. And then same kind of deal. We degrade the antigen in the phagolysosome. So that is just a fancy word for whatever whatever vesicle that I just took in from the outside world, whatever nonsense within that I pinched off from the outside space, we are just degrading whatever we just ate. So it's like digestion, kind of like that, but at the cellular level. And then MHC2 comes in and joins with this little proto-stomach that the cell has. Except it joins with something called the invariant chain. So we don't want that MHC2 to bind to anything yet until we've finished chewing our food, basically. So it has a little blocker on it in place before it can get loaded with whatever antigen we want it to be loaded with. Once it's time to load, that invariant chain gets chopped and becomes CLIP. So that's just like the butt end of the invariant chain. And then it's replaced with the antigenic peptide. So 1, 2, 1, 2, 3. The MHC2 peptide complex then moves to the surface, and we're MHC2, so this is CD4 T cell recognition. Okay, let's visualize that. Phagocytosis. So I chomp some proteins, some exogenous material from the outside world, bring it in, and then pinch it off. And now I have my phagolysosome. So the phagosome is just going to get pinched off, and then a lysosomal complex will combine. And the lysosome is really good at breaking things down. So both of those have those combine, and now I have just a pocket of enzymes to break down this protein chain into those little antigen chunks, those like 8 to 10 amino acid peptide chains. And then from my endoplasmic reticulum down here, I'm making MHC2. So this forest green here, that's MHC2. And then this whole thing is the invariant chain. So again, this is just saying, hey, don't load this just yet. We're not ready. We need to join up with the phagolysosome before any of that happens. So I make it in the ER with this blocker. It then plops off the ER and joins with that phagolysosome that had all of my antigen chunks within. Okay? So then the invariant chain, it gets broken down into CLIP and this little chain here. And then CLIP gets replaced with the antigen peptide. All right? After that, we load that MHC, that now loaded MHC2 travels in its own vesicle all the way up to the cellular membrane, where it is then met with a T cell receptor and a CD4 receptor, in this case because we are MHC2. Okay, so kind of similar to the processing pathway in MHC1, but a little bit different. Right, we have to send the MHC2 to combine with the phagosome over here, where in MHC1, we just load it up in the ER, no problem. So a little bit of a different runway there, so make sure you understand that. But all in all, very similar. We just have to load that complex with a protein that we deem to be potentially problematic. Okay. Let's talk about the genetics going on here. Now, I'm by no means a genetic professional here, but I think this is cool to mention. So this complex, it's polygenic, so multiple genes encode for MHC. For class 1, we have HLA-A, HLA-B, and HLA-C. For class 2, we have HLA-A, DP, HLA-DQ, and HLA-DR. So the combinations of those two impart what your MHC looks like. These are polymorphic, so each of those has ridiculous amounts of alleles, variants for each of those genes. There are 4,000 different types of HLA-B. That's crazy. I don't remember the numbers of the others on top of my head, but they are stark. So that means we can have a lot of combinations. A lot. So that's why HLA matching can be rather difficult. Again, I don't have the numbers off the top of my head for you, but all of those have to work out to produce the same MHC as what you have. Okay? So, something to think about. Now, it is codominant. It is codominant, meaning the maternal chunk that you get and the paternal alleles you get are expressed in each individual. So that's why you have a 25% chance of having the same HLA antigens as you do your siblings. Because you all have a chance of getting one from mom and one from dad. All right, so you have that breakdown, that typical Punnett square breakdown. Let's see if we can just do it here without me butchering. So mom 1, mom 2, dad 1, dad 2. Oh my gosh. Bear with me. Bear with me. We're okay. That looks better. Okay, so just like whatever inheritance you have, your parents also had the same deal too. They have one HLA from their mom and their father. You have one from your mom and your dad. So you just have a 25% chance if you get mom 1 and dad 1, or, one of these other combos. So that's just how the breakdown works. We have an HLA antigen from both parents, and they also had one from both parents. So that breaks down to a 25% chance that that combination happens twice. All right? Cool. So, what are the implications of having this many combinations and this many alleles? Well, this increases the likelihood that a population can present a peptide. All right? If I only had like 3 types of HLA genes, then all it takes is one virus to find a way to evade those 3 HLA genes. Right? That would be really detrimental. That means, in the age of like the bubonic plague, if all of us had no defense to that whatsoever, that's pretty damning to a populace. So that variance allows for different immune profiles, and that's good in a population. You don't want one thing to wipe you out. It ensures there you go. And it does make tissue transplant quite difficult. I think that's next lecture. And that's I'm really excited to talk about that. That's one of the inspirations behind this class. And it's a real intersection between my profession, which is anatomy, and all this cool immunology we've been talking about. It's remarkable that we are able to just pop someone's kidney out and put it in somebody else. Insane to me. But it does become quite difficult if you're not an HLA match. So again, like if you think about in TV shows, or in maybe just in real life, maybe you've heard of a story of a brother or a son or a family member donating a kidney to a family member in need. That is much more likely to happen
because of that little Punnett square action over there, that codominant expression. All right? Yeah, of course, there are potential matches in the populace. But it'd be nice to have one, just in-house, homegrown. So here's chromosome 6. You don't have to memorize this, but this is just to show you that every little thing that we've been talking about in this video is really close together, really tucked in and encoded just on this little chunk of a chromosome. I think that's just remarkable. All this science really is just magic at the end of the day, if you think about it too long. All right, this is the last slide for you. I want to talk about something called linkage disequilibrium. And this is the phenomena where certain alleles are inherited together more often than probability would dictate. Okay? So, remember I said that HLA-B has almost or around the ballpark of 400 alleles. Again, I don't know, so I'm going to fudge the numbers. I'm going to say A has like 2,600 and C has 1,800. If you did the I forget the fancy, was it preterberation? Whatever, if you do the statistical nonsense, like the factorials that get you all of the potential combinations of these, that's a very, very high number. But in nature, the number is way less. Where am I going? Let's just do this. It's way less than the equations would predict. So that must mean that there are beneficial combos that are selected for, that make more sense for biology to use. Two things to think about. The proximity on a chromosome. So the likelihood of two chunks to get inherited with one another. If A31 and B32 are close to one another, just in space, they'll get inherited together. So we'll typically see certain A and B combinations or certain B and C combinations just because they're close by. Another factor is some of these combos are just better at antigen presentation. Some of them are just the most efficient bunch. So we see some of those HLA combos more often than others due to proximity and due to just efficiency. So the pandemics and endemic infections that we see naturally select for combinations that could survive. So I mentioned the bubonic plague. Basically what that was a great filter sweeping through Europe saying which of y'all's MHC can hack it, can deal with what I have, can present this efficiently enough to get your adaptive immune system involved quick enough for this pathogen to not kill you. And was it a third of Europe did not pass that test? But the rest of them did. And there's a lot of reasons for how people survived that, but one of them would be maybe their HLA profile was a bit different and conferred more defense. Okay, so that's the idea there. So it's kind of cool if you think about it. The most prevalent HLA combinations to date are a fossil record of everything that we have had to overcome. That means that all of the MHC and the HLA genes that are expressed and alive in you must have been good enough to get your ancestors this far. Now, excuse me, in cases, like in populations that maybe didn't have exposure to something like the bubonic plague, well, maybe they weren't exposed to that pressure, and maybe they would not want to be exposed to that. I don't think anybody really would. I'm not saying that we can confer defenses to a plague through ancestral passing, but what I am saying is just that this HLA profile does get honed over time, and may give us some insight into how our immune systems have prevented and beaten voracious diseases of the past. So I have this little train choo-choo here just to resemble that sometimes, maybe like a B100 combo is just going to always get inherited with a C18 combo just because of proximity. So you have that proximity and you have the efficiency that some combos impart, meaning that not all of these combos actually exist in nature. A much less number does because of proximity and because of the pressures put on by evolution. We have been naturally selecting what HLA combos are most effective at not wiping us out as a populace. All right, so that's a little dense. I do apologize, but understanding MHC is very crucial for this unit and for immunity in general. So thank you very much for your continued attention. Have a nice day. Whoa.